Bacteria show up everywhere on the MCAT, but are you actually clear on what makes them different from everything else you've studied? From cell walls to growth curves, these tiny organisms carry a huge amount of testable detail. In this video, we'll break down the high-yield concepts you need to know structure, reproduction, and classification, so you can recognize patterns quickly and answer with confidence on test day. Bacteria are prokaryotic cells, lacking membrane-bound organelles and possessing distinct structural features such as a nucleoid region, cell wall, and sometimes capsules or flagella. This means their DNA is not enclosed in a nucleus, but instead exists in a concentrated region called the nucleoid, typically as a single circular chromosome. Unlike eukaryotic cells, bacteria do not have mitochondria, endoplasmic reticulum, or Golgi apparatus, so many of their metabolic processes occur directly in the cytoplasm or across the plasma membrane. A key structural feature is the cell wall, usually made of peptidoglycan, which provides shape and protection. Some bacteria also have an outer capsule that enhances virulence by helping them evade the immune system, a detail often tested in MCAT passages about pathogenicity. Flagella allow motility, while pili are involved in attachment and genetic exchange. Ribosomes are present but are smaller, 70S, instead of the 80S ribosomes found in eukaryotes, which is clinically relevant because many antibiotics specifically target bacterial ribosomes without affecting human cells. On the MCAT, you may be asked to compare prokaryotic and eukaryotic structures, or predict how a drug affects bacterial cells based on these differences. So focusing on what bacteria lack is just as important as what they have. Gram-positive and gram-negative bacteria are distinguished by differences in their cell wall structure, which affects staining properties and antibiotic susceptibility. Gram-positive bacteria have a thick peptidoglycan layer that retains the crystal violet stain, making them appear purple under a microscope. In contrast, gram-negative bacteria have a much thinner peptidoglycan layer and an additional outer membrane containing lipopolysaccharides, which prevents retention of the primary stain and causes them to appear pink after counterstaining. A helpful mnemonic is that gram-positive bacteria are positive for purple, both starting with the letter P. The presence of an outer membrane in gram-negative bacteria makes them more resistant to certain antibiotics and contributes to their pathogenicity, especially due to endotoxins in the lipopolysaccharide layer. On the MCAT, you might be asked to interpret staining results or predict how different bacteria respond to antibiotics like penicillin, which targets peptidoglycan synthesis. This concept also connects to immunology, because the outer membrane components of gram-negative bacteria can trigger strong immune responses, leading to symptoms such as fever and inflammation. Bacteria reproduce asexually through binary fission, a rapid process that can lead to exponential population growth under optimal conditions. In binary fission, the bacterial chromosome replicates, the cell elongates, and a septum forms to divide the cell into two genetically identical daughter cells. Because this process is relatively simple and efficient, some bacteria can double in number every 20 minutes, which leads to exponential growth described by the equation n equals n0 times 2 to the power of n, where n is the number of generations. On the MCAT, you may encounter questions that require you to calculate bacterial population growth over time, so understanding this exponential pattern is critical. However, growth does not continue indefinitely. It follows a characteristic growth curve, with lag, log, stationary, and death phases. The log phase is where rapid division occurs, while the stationary phase reflects resource limitation and waste accumulation. This concept connects to biochemistry and metabolism, as nutrient availability and environmental conditions directly influence growth rate. Additionally, understanding rapid bacterial reproduction helps explain how infections can spread quickly and why timely antibiotic treatment is important. Bacteria can exchange genetic material through horizontal gene transfer mechanisms, including transformation, transduction, and conjugation. Unlike vertical gene transfer, which occurs during reproduction, horizontal transfer allows bacteria to acquire new genes from their environment or other organisms, increasing genetic diversity. Transformation involves the uptake of free DNA from the surroundings, often from lysed cells. Transduction occurs when bacteriophages, which are viruses that infect bacteria, accidentally transfer DNA between bacterial cells. Conjugation is often considered the most direct method, involving a physical connection through a sex pilus, allowing plasmid DNA to be transferred from one bacterium to another. A useful mnemonic is TTC for transformation, transduction, and conjugation. On the MCAT, this concept frequently appears in questions about antibiotic resistance, as resistance genes are often carried on plasmids and spread rapidly through bacterial populations via conjugation. This also ties into evolution and natural selection, as bacteria with advantageous traits are more likely to survive and proliferate in selective environments, such as those containing antibiotics. Bacterial metabolism and oxygen requirements vary widely, with classifications such as obligate aerobes, obligate anaerobes, and facultative anaerobes, reflecting their energy production strategies. Obligate aerobes require oxygen for survival because they rely on aerobic respiration to generate ATP efficiently. Obligate anaerobes, on the other hand, cannot tolerate oxygen and may be harmed by reactive oxygen species, so they rely on fermentation or anaerobic respiration. Facultative anaerobes are versatile and can switch between aerobic and anaerobic pathways depending on environmental conditions. A helpful way to remember this is that facultative organisms are flexible. On the MCAT, you might be asked to predict bacterial growth in different environments or interpret experimental data based on oxygen availability. This concept connects closely to biochemistry topics such as the electron transport chain, fermentation pathways, and ATP yield. For example, aerobic respiration produces significantly more ATP than fermentation, which explains why obligate aerobes depend on oxygen. Understanding these metabolic distinctions also helps explain where certain bacteria are found in the body or environment, such as anaerobic bacteria thriving in low oxygen regions like the gut,